Welcome back, everybody. We're going to build on what we learned on the endocrine system by moving over to the reproductive system. And we will relate what we did with endocrinology to this system. We're going to start off in this mini lecture with the male reproductive system. And then the last mini lecture in this series will deal with the female reproductive system. There's going to be some anatomy review in this lecture, but as always, I'm going to try and relate the anatomy to a physiological function. So you learned the anatomy of the scrotum in anatomy, and this slide just shows you basically that the physiological function of the scrotum is to form a supportive structure for the testes. It is basically there to support the testes and also, more importantly, to suspend it outside of the body's internal cavity. The fact that it's not found in the pelvic cavity means that the internal temperature of the scrotum is slightly cooler than that of the pelvis. And this is important because the process of making sperm, or what we'll see is called spermatogenesis, is not as efficient at the internal temperature of the pelvis. So it's not very efficient at 30 degrees Celsius. But having the testes supported away from the pelvis and having a slightly cooler temperature means that spermatogenesis can be optimized. Each testes is associated with some muscles. The first muscle that we'll talk about is called the cremaster muscle. This is actually voluntarily controlled skeletal muscle. It is a continuation of the internal oblique. The testes develop just underneath the kidneys, and as gestation continues, they begin their descent through into the pelvic cavity and into the forming scrotal sac. And as the testes descend into the scrotum, they take the internal oblique with them, and this becomes the cremaster muscle. Now, in addition to this skeletal muscle, there is also built into the wall of the scrotum, an involuntarily controlled smooth muscle called the dartos muscle. Together, these two muscles, when they contract, can lift the scrotal sac with the two testes closer to the body. So if the temperature is a little cooler than normal in the scrotal sac and spermatogenesis efficiency is being impacted, by contracting these muscles and bringing the scrotum closer to the body, you can warm up the internal temperature of the scrotal sac, thus establishing more efficient spermatogenesis. The actual term cryptorchidism is associated with the failure of a testes to descend into the forming scrotal sac. It's not very often seen in full-term infants, maybe 3 to 5% of full-term infants, and in those infants, usually the testes will descend within a month or so. But it's seen in a full third of premature infants, just simply because the scrotal sac hasn't finished developing fully. If untreated, it can result in sterility, but usually the course of action is just to wait, usually a few months to see if the testes descend on their own normally. If not, it can easily be rectified with surgery. As I mentioned in the previous slide, the testes do develop internally, just underneath the kidneys, and they will descend through the pelvic cavity and through the inguinal canal during the latter half of the 7th month of gestation. As they descend into the forming scrotal sac, they will take a portion of the internal oblique with them. This forms the cremaster muscle. But they also take part of the serous layer of the peritoneal membrane. So they will actually take part of the parietal peritoneum with them. And this will form a protective layer around the testes called tunica vaginalis. The layer inside of that is called the tunica albuginea, and it actually divides the testes up into lobules. So if you wanted to look at it from the anatomical perspective, you would have the skin of the scrotum, the dartos, the cremaster, the tunica vaginalis, and then, like a saran wrap layer covering the testes and dividing it into little lobules, you would have the tunica albuginea. And the tunica albuginea actually divides the testes in anywhere from about 200 to 300 lobules. And now we're going to take a look and see what's inside each of those lobules. Each lobule of the testes is going to contain anywhere from 1 to 3 tubes. These are coiled tubes, and they are known as seminiferous tubules. The cells found within the seminiferous tubules undergo meiosis to produce sperm. And they are lined actually with a specialized type of stem cell, or also known as a germ cell, which is called a spermatogenic cell. This spermatogenic cell would be located at the periphery. So this layer right here that I'll highlight here in green, the most peripheral or superficial layer of that seminiferous tubule would be lined with spermatogenic cells. The spermatogenic cells or germ cells are diploid. They will undergo meiosis and they will shed into the hollow lumen of that tubule the haploid spermatids. So the site of spermatogenesis will be these hollow tubes called seminiferous tubules. The deepest layers, as I mentioned, contain the type of cell called a spermatogenic cell, but they also contain another cell which supports spermatogenesis, and this second type of cell is called a Sertoli cell. So in the most superficial layers, or I guess I should say the deepest layers, that layer I outlined in green, you will find two types of cells. The diploid spermatogenic cells, which will undergo meiosis to produce sperm, and cells known as Sertoli cells. The last thing I'll say is that the spermatogenic cells are often called spermatogonia or spermatogonium singular. So these sperm stem cells are known as either spermatogonia or spermatogenic cells. This is a really nice picture here that shows you the location of these spermatogenic cells, or these sperm stem cells right here, undergoing meiosis and giving rise to the spermatids, which will then form into the sperm. And then you can see right here, this cell right here that I'm going to highlight in pink, this very big cell, that is a Sertoli cell. We'll learn a little bit more about these Sertoli cells in a subsequent slide. The Sertoli cells surround the cells that are undergoing spermatogenesis. They've been known as nurse cells because they secrete a lot of supportive chemicals that support and help mediate spermatogenesis. A lot of textbooks will also call these Sertoli cells sustenacular cells. They are very, very large cells. I am going to outline one Sertoli cell right now. This thing all here that I'm outlining in purple, this really big, oddly shaped cell, that is a Sertoli cell. This right here is the second one that I'm outlining. And you notice what is sandwiched in between? The actual spermatogonium or spermatogenic cell, which is undergoing meiosis and forming the spermatids, which will become the sperm. They secrete a wide variety of growth factors that help mediate this process of spermatogenesis. They also consume the unneeded portions of the spermatozoa. One of the secretions that they produce creates something which is known as a blood-testes barrier. There is an actual protection in the testes against the man's immune system, and it is one of the secretions of the Sertoli cells. So think of the Sertoli cells making up the seminiferous tubules, part of the seminiferous tubules, and then protecting the seminiferous tubules from attack by their own immune system. It creates a privileged immune environment, which is fairly similar to the blood-brain barrier. This slide outlines additional secretions that are made by the Sertoli cells. So there are all kinds of cytokines and growth factors that actually are secreted by the Sertoli cells. Let's take a look at a few of these substances. I'm going to start off with anti-Müllerian hormone. This is secreted during the early stages of fetal development. Even before the male reproductive system forms, Sertoli cells appear and begin to secrete anti-Müllerian hormone. This is to determine the development of male reproductive structures. This hormone prevents the development of the Müllerian ducts, which will become the female reproductive system. In addition to anti-Müllerian hormone, there are two reproductive hormones that are going to be made. They're made and secreted after puberty. So after the male reproductive system forms, the Sertoli cells of the testes will secrete inhibins and activins. We'll see what inhibin does more specifically. You've probably already figured out that inhibin probably inhibits something. We're going to see that it's going to work together. These two hormones will work with one another to regulate follicle-stimulating hormone secretion and its action in the reproductive system. I think we'll concentrate more, though, on inhibin because that's a little bit better described in the literature. Finally, Sertoli cells also secrete something called androgen binding protein. This is going to play a really important role in helping spermatogenesis and the maturation of the forming sperm. It basically acts to bind and concentrate testosterone in the seminiferous tubule so that these spermatogenic cells can undergo more efficient meiosis. There is another cell of the testes that I want to tell you about now. They are called the Leydig cells or the interstitial cells. These are cells which are found in between the seminiferous tubules. So in this diagram right here, we would have a portion of a seminiferous tubule here. You might have another seminiferous tubule maybe here. And then in between, you've got these guys right here. These cells that are found in between seminiferous tubules, right next to, coincidentally, capillaries of the vascular system. These Leydig cells can be found. They secrete one thing and one thing only, androgens. They produce the androgen testosterone. And testosterone is vitally important for efficient spermatogenesis. By the way, Leydig cells and Sertoli cells can be reproduced in large enough quantities to produce tumors. So both Leydig cells and Sertoli cells can be a type of testicular cancer. I want to go back to this idea that Sertoli cells can create a privileged immune environment within the testes. There have been studies that have taken Sertoli cells, and they can be transplanted into an outside site, a site outside of the reproductive system, known as an ectopic site. So they can take these cells and they can implant them into, let's say, the skin of a pig. So you're going to take pig Sertoli cells, and you're going to put these Sertoli cells back in the pig somewhere other than the pig's reproductive system. Let's implant these cells into the skin of the pig on its back. Studies then took skin cells from a dog and created a graft right in the center of that ring of Sertoli cells. And the pig's immune system was incapable of recognizing those dog skin cells as being foreign, and the pig's immune system could not attack those foreign cells. The Sertoli cells implanted into the skin of the pig created a privileged immune environment in which the dog skin cells could survive. This could have really important ramifications for organ transplants, because if you can transplant an organ into a host and surround that transplanted organ with live, viable, reproducing and secreting Sertoli cells, there wouldn't be the need for any tumor rejection drugs. You can see here in this slide, I could tell you that another study they did this with canine pancreatic cells. Not only was the pig's immune system incapable of recognizing those dog cells, but those canine pancreatic cells still maintained their function and produced insulin. Again, this opens up a whole broad range of transplantation medicine that maybe we won't need these powerful anti-rejection drugs that you would have to take day after day for the rest of your life. So it could be a really exciting advance in transplant medicine. Remember, the other type of cell found within the seminiferous tubule is the actual sperm cell called a spermatogonium or spermatogonia plural. And another word for a spermatogonia, it will be the spermatogenic cells. Spermatogenic cells actually form from what are called primordial germ cells. These primordial germ cells are derived from an embryonic tissue called the yolk sac. They travel in the forming embryo to where the testes are going to form, and they transform themselves into spermatogonia or spermatogenic cells. And these spermatogenic cells will remain dormant in the testes until puberty starts. And then every single day for the rest of that male's life, the spermatogonia will undergo spermatogenesis. Now, in order for the male to maintain enough spermatogonia so that it lasts the rest of his life, the first thing these spermatogonia have to do is they have to undergo a round of mitosis before they start meiosis. The idea is that a spermatogonium splits into two identical spermatogonia. One spermatogonium is going to undergo spermatogenesis, in other words, meiosis, and form into 4 sperm. The other spermatogonium stays a spermatogonium stem cell. The next day, it splits to form again 2 spermatogonia. One undergoes meiosis, one stays as a stem cell, and then that keeps dividing and dividing and dividing. That is basically self-renewal, isn't it? You split, create 2 identical copies, keep one copy as a stem cell, let the other one undergo meiosis. This means that the male will have enough spermatogonia from the first day of puberty until the day he dies. We're going to take a look at this process, but I will tell you that it begins at the edges of the seminiferous tubules. The dividing cells, the meiotic dividing cells, are going to proceed towards the lumen of the seminiferous tubule, and that is what the spermatozoa, or the sperm, will be shed off into the lumen of the seminiferous tubule. It's going to take about 60 to 75 days to start from mitosis and go all the way through meiosis 1 and meiosis 2. And one other interesting fact, I don't know if you can see in this diagram, but each cell here undergoing spermatogenesis, they are physically linked to one another, so that all spermatogonia will enter into meiosis 1 at the same time. And then those meiotic 1 cells will enter into meiosis 2 at the same time. So you have some coordination, where those cells that are undergoing meiosis do it in a coordinated fashion all at the same time. This results in the production of anywhere from 2 to 300 million sperm each day. All right, let's assume we've undergone mitosis. We've taken that 1 spermatogonium, we've split it into 2. So one is going to remain in the deep layers near the basement membrane of the seminiferous tubule. But the second daughter cell, and I know it's weird how we call it a daughter cell, is going to detach from the basement membrane and enter into meiosis. The first thing that spermatogonium has to do is it has to transform itself into a cell called a primary spermatocyte. The germ cells which will give rise to these sperm stem cells are diploid. The spermatogonium stem cells that will give rise to sperm, they're diploid. The very first cell of meiosis, the primary spermatocyte, it's diploid. We're going to undergo meiosis 1. At the end of meiosis 1, you are going to have 2 haploid secondary spermatocytes. Now, the chromosomes in those secondary spermatocytes are going to be this kind. They're going to be the duplicated chromosome. But instead of having 2 sets, each cell is only going to have 1 set. You are now going to enter into meiosis 2, in which case you are going to end up with 4 haploid cells called spermatids. Each one of these spermatids will have the chromatid chromosome like that. So they'll have, in our case of humans, 23 of these. Now you'll notice this isn't the end of spermatogenesis. The very last stage of spermatogenesis is the transformation of these cells which don't like sperm, they don't look like sperm, into cells that look like sperm. So that very last stage is called spermiogenesis, which means creation of sperm. It is the physical development of cells called spermatids into spermatozoa, or what you can just call sperm. You can see in this picture that the spermatids are spherical. They are going to transform themselves into a long cell with a head and a tail, or what you could call a flagellum. Typical human male will make 300 million of these each day. A sperm is 60 microns long, and it will contain the following parts: an acrosome, a head, and a tail, or what you can call a flagellum. The head contains a single nucleus with 23 highly condensed chromatid chromosomes. So again, the chromosome looks like this if I were to do it like that. The chromatid chromosome. Covering the tip of the sperm is a very special organelle called an acrosome. So this covers the head of the sperm. It is only found in sperm, and it is an organelle that contains numerous digestive enzymes. These enzymes are there for one purpose: to dissolve a protective barrier that surrounds the egg. And this protective barrier we're going to see in the female reproductive system. It is called the zona pellucida. It is a thick protective layer of proteins and sugars that surrounds the ovulated oocyte and will surround the egg. And it actually is what the sperm has to dig itself through in order to contact the plasma membrane and get internalized into the egg. So some of the enzymes that are found in the acrosome are there to digest like the sugars, the hyaluronan sugar of the zona pellucida, and there's all kinds of proteases to digest the proteins of the zona pellucida. The flagellum is known as the tail. It's made up of several pieces like a neck. The neck contains a centriole. The centriole is going to be really important because it is going to be part of the centriole of the fertilized egg. So it's going to be one of the two centrioles that the fertilized egg makes when it undergoes its very first round of mitosis. The middle piece is a bunch of mitochondria. These flagella have got to move, and the mitochondria make ATP in order to create the undulating or wave-like motion of the flagella. The actual majority of the flagella is called the principal piece, and then there's a little teeny tiny thin section called the terminal portion or the end piece. The hormonal control of the testes involves the hypothalamic hormone called gonadotropin releasing hormone, the two gonadotropins, follicle stimulating hormone and luteinizing hormone, and then there are going to be the reproductive hormones testosterone and inhibin. Now, out of those hormones, GnRH, LH, FSH, and inhibin are also going to have very similar roles in the female reproductive system. Gonadotropin releasing hormone is going to come from, again, the neurosecretory cells of the hypothalamus. Those neurons that are going to make this hormone, their axons are going to end at the hypophyseal portal system. So the GnRH is going to get secreted into that portal system. It's going to diffuse via the bloodstream down to cells in the anterior pituitary called the gonadotrophs. So when the gonadotrophs bind GnRH, they will begin to produce the two gonadotropins, FSH and LH. GnRH release and FSH and LH release are going to be subject to negative feedback systems. For example, increased levels of testosterone can feed back to the hypothalamus and the anterior pituitary, and they can shut off GnRH release and luteinizing hormone action. Let's start off with follicle stimulating hormone. This is a hormone that's actually named for its role in the female reproductive system, in that it causes the development of follicles in the female's ovary. But in the male testes, it actually indirectly stimulates spermatogenesis. It acts synergistically with testosterone on the Sertoli cells. This means that by itself, FSH can stimulate Sertoli cells. By itself, testosterone can stimulate Sertoli cells. But you put them together and you get a synergistic or additive effect. And really, to be honest, if you want effective spermatogenesis, you need both FSH and testosterone. Testosterone actually stimulates the production of FSH receptors on the Sertoli cell so that FSH can bind the Sertoli cell and stimulate the Sertoli cell's secretion of androgen binding protein into the fluid of the seminiferous tubule tissue. Androgen binding hormone does exactly what its name implies. It's going to bind testosterone to keep its concentration high within the seminiferous tubule. And this is important because not only is testosterone needed for Sertoli cell action, but testosterone stimulates the final stages of spermatogenesis. FSH release is inhibited by the release of inhibin. Inhibin is produced by Sertoli cells. So while Sertoli cells are making things like androgen binding protein, they're also making inhibin. And when inhibin levels climb high enough, the inhibin will negatively feed back to the anterior pituitary and, just feeds back to the anterior pituitary. I've learned that a lot of figures showing inhibin's feedback to the hypothalamus may actually be incorrect. So I'm going to err on the side of caution and just tell you that FSH is going to negatively feed back to the gonadotrophs of the anterior pituitary. That's going to shut off FSH. It's also going to shut off LH production and release. Luteinizing hormone is named for its role in the female reproductive system in that it causes the development of the corpus luteum, and the corpus luteum makes estrogen and progesterone. So luteinizing hormone is a hormone that's important for the reproductive system's production of reproductive hormones. So in the male, luteinizing hormone, coming from the anterior pituitary, stimulates the Leydig cells of the testes, telling them to make testosterone and the other androgens like 5-DHT. Testosterone is made from cholesterol in the testes. The Leydig cells will take cholesterol out of the bloodstream, will modify it and convert it into testosterone. And testosterone, when their levels get to be high enough, they will negatively feed back to the anterior pituitary and the hypothalamus, thus suppressing gonadotropin-releasing hormone synthesis and suppressing luteinizing hormone synthesis, and also FSH. In some targets, like the prostate gland and the seminal vesicles, the testosterone that's made by the Leydig cells gets converted into dihydrotestosterone, which is known as DHT. Luteinizing hormone results in the production of testosterone from the Leydig cells. Now, testosterone's target is going to be the Sertoli cells of the seminiferous tubules. And the testosterone actually enters into the Sertoli cell and stimulates the Sertoli cell's expression of the FSH receptor. And I know it's really hard to see this, but I'm going to circle that right there. That actually, I believe, is the testosterone entering into the cell, entering into the nucleus, and causing the cell to express the FSH receptor. And you can see that little rectangle there, that's the testosterone binding the FSH receptor. And what results is androgen binding protein secretion by the stimulated Sertoli cell. In your Canvas page, I kind of rework this figure because, again, it's not very clear by this figure exactly what's going on. So I kind of reworked it in the reading on the male reproductive system. So again, let's go over it again. Testosterone enters into the Sertoli cell, that really big Sertoli cell. It moves into the nucleus. It physically binds a very specific sequence of DNA in the Sertoli cell's nucleus. And in response, that Sertoli cell begins to express and produce the FSH receptor. That FSH receptor gets expressed on the surface of the Sertoli cell so that now FSH, that little rectangle there, can bind the receptor. This is going to trigger the Sertoli cell to produce androgen binding protein. Androgen binding protein's job is to bind testosterone within the fluid of the seminiferous tubule to prevent its escape from the seminiferous tubule. And this increased level of testosterone within the seminiferous tubule is going to stimulate the meiotic division of spermatogenic cells. It is going to stimulate the latter stages of spermatogenesis. Luteinizing hormone will also promote the conversion of testosterone into dihydrotestosterone, which is also known as 5-DHT. There are several kinds of androgens, but the major types of androgens are testosterone and 5-dihydrotestosterone, or you can just call it 5-DHT, or if you want DHT for short. Both of these types of testosterone bind to the same testosterone receptors. The receptors for testosterone are found inside the nucleus of the targeted cells. So, for example, the testosterone receptor is found in the nucleus of the Sertoli cell. Why are they not expressed out on the surface? Simple. Testosterone and DHT are lipid-soluble hormones. They actually can get through the plasma membrane and into the nucleus, piece of cake, where they can find their receptor. And their job is to bind specific sequences of DNA in the targeted cell's nucleus. So in the case of a Sertoli cell, the testosterone receptor complex binds a region in the promoter just in front of the FSH receptor gene. If you were to look in a muscle cell, testosterone and its receptor would bind to sequences in the promoters of muscle protein genes. So, for example, you might get increased expression of myosin as a result of testosterone stimulation, or actin. That's the effects we see in adults. It plays a role in reproduction and spermatogenesis. It plays a role in an anabolism in the production of, say, for example, muscle and bone proteins. But testosterone has a really important role in prenatal development of the male reproductive system. Now, I'm going to say, first of all, when I say male reproductive system, I'm talking about the structures of the testes, the prostate, the seminal vesicles, even the external genitalia. You notice I'm going to use the word male in terms of genetic male, having an XY chromosome and having those reproductive structures. So I'm not using male in the sense of gender. I'm using male in the sense of genetics and physiology. Testosterone is going to specifically stimulate the development of the epididymis, the vas deferens, the ejaculatory duct, and the seminal vesicles. And if you don't remember what those are, go back to your anatomy notes and look those up. It's also converted in the brain to estrogens and is responsible for the development of certain brain regions in males. The job of 5-DHT, or DHT, is to stimulate the development of the external genitalia. This development begins around the 8th week. The external genitalia come from a structure in both males and females called the genital tubercle. This is comprised of a urethral groove and 2 labiosacral swellings. And in males, the genital tubercle will elongate into the penis, and the labiosacral swellings become the scrotum. So this slide summarizes probably the roles of testosterone that you're more familiar with. The stimulation of anabolism. It stimulates protein synthesis, specifically in bone and muscle, so that males are larger than females. It's very important in the development of sexual function, male sexual behavior, spermatogenesis. But it also has important roles in the libido in both males and females. Although in females, probably the DHEA androgen coming from the adrenal cortex will also play a role, whereas DHEA, that androgen, has virtually no role in libido or in reproductive function in males. And then finally, testosterone is responsible for the development of male secondary sex characteristics. Now, if we were in class right now, I would ask you what some of those secondary sex characteristics are. And I think the first thing that you would probably come up with is increased muscle mass. Most students either say increased muscle or hair, and they would be correct. Because of its anabolic role, testosterone leads to increased muscle mass and increased bone mass. As I just said, most males are bigger than females. Also, most males will have more body hair. Ironically enough, DHT stimulates the production of hair on the body, but it actually prevents the growth of follicles on the scalp. So testosterone causes body hair, but also causes male pattern baldness. And then a few students usually come up with a deep voice. Males, because testosterone increases the size of the larynx and increases the size of the vocal cords, results in the Adam's apple and a lower voice. What are these secondary sex characteristics for? They are subtle advertisements to a female, saying to the female that you are a reproductively healthy male, you will have no problem in impregnating her, getting her pregnant, and you will have no problem supporting her until the offspring is born. So it just means basically, hey, look at me, you want to get pregnant by me. Remember that testosterone operates through your conventional negative feedback mechanism. Based on blood levels of testosterone, you will either inhibit the hypothalamus and anterior pituitary, or you can actually remove that inhibition, and then that system, that hypothalamus pituitary axis system, turns back on. Here's an anatomy slide which is outlining the reproductive ducts, the vas deferens, the urethra, and also the epididymis. Physiologically, the epididymis is the site of sperm maturation. Sperm actually acquire mobility within about 14 days. The cells lining the epididymis also help propel the sperm into the vas deferens. And the vas deferens is a small, smooth muscle-lined tube that helps propel the sperm up into the urethra. The connection of vas deferens to the urethra is the section known as the ejaculatory duct. It passes through the prostate and it will terminate in the urethra. And remember from anatomy that the urethra is divided into 3 sections. The portion which runs through the prostate is called the prostatic urethra. The numerous ducts that will come from the prostate connect to the prostatic urethra. There's a membranous urethra which is found between the prostate and the erectile tissue of the penis. The bulbourethral glands actually have a duct and connect into the membranous urethra. But the bulk of the urethra is the spongy urethra, which is the portion which runs through the spongy erectile tissue of the penis. The last thing I'll remind you is that, remember, the ejaculatory duct is actually not only where the vas deferens connects onto the urethra, but it's actually where the seminal vesicle connects to the urethra. So if you remember from your anatomy, the ejaculatory duct is actually the shared duct between the vas deferens and the seminal vesicle. So the seminal vesicles actually contribute their secretions to the prostatic urethra via this ejaculatory duct. Again, I'm going to encourage you guys to go back to your anatomy notes and take a look at these things because they do have relations to the physiology of these structures. The male reproductive glands you learned in anatomy were the seminal vesicles, the prostate gland, and the 2 bulbourethral glands. Collectively, what these glands do is they produce the fluids that combine with sperm to make semen. The vas deferens contains sperm, the urethra contains semen. That's as simple as I can put it. Semen is an alkaline fluid. It contains numerous chemicals and growth factors that help activate the sperm cells so that they acquire the ability to swim through the female reproductive tract. Let's start with the prostate. It surrounds the urethra. It actually forms as an outgrowth of the urethra along with the bulbourethral glands. So urethra, prostate, and bulbourethral glands all have the same embryologic origin. Its secretion is thin and milky. It enhances sperm motility, and it helps neutralize the acidity of vaginal fluid. The inside fluid that is found inside the vagina is very inhospitable to sperm. It is thick and very mucus-like, and it is somewhat acidic because the female reproductive system is going to do everything in her power not to allow a pregnancy to take place. Believe it or not, pregnancy is a very vulnerable, dangerous time in a female's life. So the female actually tries to minimize the number of times she can get pregnant. And she does so by changing the actual physical and chemical environment of the vaginal fluid. So the prostate secretions try and counteract this by helping to neutralize the acidity of vaginal fluid. 2 seminal vesicles are found on the back of the bladder. Again, as I mentioned, they help share a duct. They share a portion of the ejaculatory duct with the ductus deferens or the vas deferens. So both the sperm coming from the vas deferens and the secretions of the seminal vesicles get dumped into the prostatic urethra via the ejaculatory duct. The secretions of the seminal vesicles is an alkaline fluid, again, to help counteract the acidity of the vaginal fluid. But seminal vesicle secretions also contain sugars so that the mitochondria of the sperm can make ATP. I believe most of these sugars are fructose, and they also contain the hormones known as prostaglandins. This class of hormones called prostaglandins is known to promote smooth muscle contraction, and the thought is that the prostaglandins found in the seminal vesicles stimulates uterine smooth muscle contractions to help drive the sperm further up the uterus towards the 2 fallopian tubes. Finally, we have the bulbourethral glands. These are 2 glands behind the prostate. They're not shown here, but if I wanted to actually put like the urethra here, the bulbourethral glands would be located about here. Bulbourethral glands, bug. Also known as Cowper glands. Their secretions is a thin, slippery fluid which will lubricate the tip of the penis so that it can be inserted into the vaginal canal. The physiological function of the penis is to convey urine and semen. Now here's again the anatomy of it. I will remind you that the body of the penis is made up of two types of erectile tissue, the corpus cavernosum and the corpus spongiosum. These tissues actually fill with blood. This blood is from branches off of the testicular artery. And by filling these spaces, they actually lengthen the penis, widen it, and harden it so that it can be inserted into the vaginal canal, so that the semen can be delivered through ejaculation. Gonads develop during the 5th week of gestation. At the 5th week of gestation, there are two sets of ducts, the Wolffian ducts and the Mullerian ducts. Now, Wolffian ducts, if they are allowed to develop, become the reproductive structures of the male, and the Mullerian ducts, if they are allowed to develop, become the reproductive structures of the female. And again, I'm referring to males as being XY chromosomes. Physiologically, they are going to have functional testes. The female is going to have two X chromosomes. Physiologically, they are going to have functional ovaries. So really, the embryo has the potential at that 5th week, when those two ducts are there, to develop into either sex. But quote unquote, what I say, maleness, is determined by a gene called SRY, the sex determining region of the Y chromosome. What does the SRY gene become expressed as? Well, when it's transcribed and translated, you end up with a protein called TDF, testes determining factor. Testes determining factor induces the differentiation of Sertoli cells from the cells of the Wolffian ducts. And once they begin to appear, these Sertoli cells are going to secrete anti-Mullerian hormone. This hormone is going to result in apoptosis of the Mullerian ducts, thereby inhibiting the development of the female reproductive structures, and this then allows the development of the male reproductive structures. So really, the default sex, or the default reproductive structures, is female. In response to another hormone being produced by the embryo called human chorionic gonadotropin hormone, HCG, you now have the appearance of Leydig cells from the cells of the Wolffian ducts. Once they begin to appear, they start synthesizing their testosterone, and this influences the development of male reproductive structures from the remaining parts of the Wolffian ducts. Specifically things like the epididymis and the vas deferens, the ejaculatory duct, the seminal vesicles.